Hey everybody, in this video we are going to talk about sex determination in humans, specifically genetics sex determination. So determination, okay, so we are not talking about gender, right? So that's a different subject altogether that I know it's not part of this course. So we're talking about genetic sex. And what we mean by genetic sex, well, essentially what we would consider genetic sex would be those individuals that develop testes. So we'll consider individuals that develop testes as males, and at least with respect to genetic sex, and females will develop ovaries. So we are interested in learning about what determines whether an individual develops testes or ovaries with respect to genetics. So we will take a look at the sex chromosomes in humans. And when we say, what are sex chromosomes? Well, those are the chromosomes that determine sex. So in humans, we have X and Y chromosomes and X chromosomes. So two combinations that are typical in humans. In the X and Y, those individuals develop testes. In XX, these individuals develop ovaries, well, at least during embryogenesis. So our first question is why? Right? Why does, I say a zygote that starts out as XY, why does that zygote, through embryogenesis eventually develop testes? And why does an XX develop ovaries? So we could think of a number of possible reasons, right? So is it, say, one X, just having one X chromosome? Let's take a look at this XY. Does having one X chromosome cause testes to develop? And it turns out that's the way it works in flies. So it's not the Y chromosome in flies. It's the fact that males only have one X and females have two X's. Well, at least in Drosophila melanogaster. So another possibility, is it the presence of the Y? Is it the Y chromosome that causes, an embryo to develop testes? And turns out it is the Y. It is the Y, and we're going to see why in this video. Get rid of this here, this here. So let's take a look at the Y chromosome. So here is a simple diagram of the Y chromosome. Now, this is the P arm, this is the Q arm. Remember, the P arm is always shorter than the Q arm. So these regions at the tips, these are called the pseudo-autosomal regions. So PAR. So this is a PAR region up here, there's one down here, and these are the things that allow the Y chromosome to pair up with the X chromosome during meiosis, because they're similar, these sequences are similar to the sequences found on the X chromosome. Now all this stuff in between the pseudo-autosomal region, all that is specific to the Y. Specific to Y, so it's unique to the Y, and it's not found on any other chromosome, including the X chromosome. Now this region right here, we're going to call this is the euchromatic region. Now we talked about euchromatin in the past. Euchromatic right here. Euchromatin is chromatin that is, say, loosely organized. The nucleosomes aren't so compact, right? And so scientists tend to think euchromatic regions Well, the genes in this region right here are more easily expressed or transcribed than they are from heterochromatic regions. So this region down here, well, this is going to be the heterochromatic region. And remember, heterochromatin is a form of chromatin where the nucleosomes are compact. So the genes within heterochromatin are difficult to transcribe. Now the Y chromosome only has 50 to 75 genes, and I don't know why we don't have a definitive number here. But so we're going to leave it at 50 to 75 genes, and maybe all the genes haven't been characterized yet, or maybe some genes, you know, scientists can't tell if they're functional or if they're relics. I'm not sure why, but what we do know is that there's a very small number, relatively small number of genes on the Y chromosome. And I believe most of them are found in the heterochromatic region, which would make sense because that's the region where it's easier to transcribe genes from or within. Now this piece right here, this is where we're going to find a gene called SRY for sex determining region of the Y. So I'm guessing at first it was a region, but now we know there's a gene, and the gene there is called SRY, and that is a gene that encodes a transcriptional activator. And you can probably guess what this transcriptional activator does. It triggers development of testes. Okay, so this one gene right here can trigger development of testes during embryogenesis. So how does that happen? So that's essentially why the Y chromosome controls sex determination in humans. So about 5 weeks after conception, this is in humans now, you know, we're going to have, let's say, the embryo looks something like this. If you look at images of embryos at this stage, it'll look something like this. Now, I'm imagining this might be the head region. This might be hindgut, maybe. Not really sure. But what I want to point out right here is somewhere in here, on each side, it's going to be this tissue that is called the genital ridge. And there are two of them, so there are two genital ridges, say on either side of this little embryo right here. Now we can also call these bipotential gonads, because what we think is that at this point, at 5 weeks, this embryo right here can develop either testes or ovaries. So it can follow the male developmental pattern or the female developmental pattern. And it's the genital ridges that can, you know, what they have is two types of tissue called the medulla and the cortex. Now the medulla is what develops into testes, and the cortex will develop into ovaries. The medulla of one genital ridge will develop into a testes, while the cortex of one will develop into an ovary. And then we'll have pairs of testes or ovaries. So at about 5 weeks, now this is when the signal from SRY, either it begins or maybe it's not recognized until 5 weeks. But if SRY, the protein product of the SRY gene, that transcriptional activator, is present at 5 weeks, the medulla develops into testes. If SRY is not present If that gene is not present and this protein product is not present, that transcriptional activator is not present, what happens is the cortex will develop into ovaries. So we can consider ovary development as the default condition. So the ovaries are going to develop unless this SRY signal is present at 5 weeks. Now, if something goes wrong with the signaling, what we can get is these things called streak gonads. So these are gonads that don't develop, gonads being either testes or ovaries, that they don't fully develop and they're immature and they're non-functional. So if something goes wrong after this point right here during development, we've got these streak gonads, and there are some conditions that involve formation of neither testes or ovaries, but formation of streak gonads. So how can something go wrong? Okay, so we have two ways that we'll cover. And the first one is going to be non-disjunction during meiosis. So non-disjunction, what is that? So non-disjunction, and you know, I've been looking for a way to introduce this term into the first part of the course. But you know, the way I've done things in the first part of the course, we really haven't had time to talk about this or about major chromosome rearrangements. But so I'm going to introduce this here. So non-disjunction is failure of chromosomes to segregate during meiosis. Segregate, maybe separate. Segregate, separate. I might like separate better here. Those terms are so similar. And maybe it's best for me to just show you what happens. So let's take, say, oogenesis. So this thing we talked about, this process, oogenesis and spermatogenesis, that we introduced, I think, in the second lecture of this course, turns out to be really important, right? We come back to it throughout the semester over and over again. So let's take the primary oocyte here. And during oogenesis, there will be 2 X chromosomes, if we're talking about human females. And let's say, oh, I should have diagrammed these closer together, huh? So these typically separate, right, during the first meiosis. So this is the primary oocyte that goes through the first meiosis. That's an X chromosome, that's an X chromosome. See, this is the nucleus. Sometimes they don't segregate to the products of meiosis 1. This is going to be the first polar body. Say first polar, not primary polar body, first polar body. And right here is the secondary oocyte. Now, let's say both X's went this way. So now we have 4 X chromatids, 1, 2, 3, 4, 2 chromosomes, but 4 chromatids in the secondary oocyte, 0 X chromosomes or chromatids over here in the polar body. Now remember, this is a single chromosome, this is a single chromosome, these are 2 chromatids here. This is a chromosome with 1 chromatid. Chromatid. So, okay, so we have non-disjunction occurring, and we have too many X chromosomes over here, or at least more than normal, right? Now, this has got to go through another meiotic division, right? And okay, let's say we have the second polar body here. And let's say they separate normally here, and you know, how would they align during the second meiosis, right? They're going to align one on top of another. Even though they're both X chromosomes, there's no pairing during the second meiotic division. So the align one on top of the other during metaphase. And keep in mind, all the other chromosomes are here. We're just looking at the X chromosomes in this diagram. So let's say this is the second polar body, and there are 2 X chromatids here because they segregated normally. So, right, if they're lined up like here, we're going to have 2 X chromatids inside the polar body, but they're going to be lost. And over here, we have 2 X chromatids. Okay, and we also have, what, the 22 other chromosomes, right? The 22 autosomes. Let's say they all separated normally during both stages of meiosis. So this ootid right here is normal except for 1 extra X chromosome. Now, the same thing can happen during spermatogenesis. So let's call this the primary spermatocyte. And let me have a diagram the X chromosome here, and I'll make the Y a little smaller. We'll find out it's about a third of the size of the X chromosome. So let's say non-disjunction occurs during meiosis 1. Non-disjunction here. Okay, non-disjunction occurred up here, right? 2, non-disjunction. Now, so in this guy over here, this secondary spermatocyte, we have 0 sex chromosomes over here because both the X and the Y, let's say, went this direction. So now what we have is the X up here and the Y right here. And all the other chromosomes are here. So the 22 other chromosomes are here also. They're also over here because those, let's say, segregated normally. Now that through meiosis 2, let's say disjunction occurs normally. So we have spermatids, each with 1 X chromosome and 1 Y chromosome, right? So if these are the chromatids up here, they separate normally. 1 X goes this way, the other X goes this way. Then 1 Y goes this way, right there, and another Y goes this way. So we end up, let's say these are the spermatids, and they're going to mature into spermatozoa. Each has an X chromosome and a Y chromosome. And you know, I just went over 2 examples here, but you can get many different combinations of X chromosomes and Y chromosomes in the spermatids during spermatogenesis. Now keep in mind, you got to follow those all the way through too. And these are going to be sperm with no sex chromosomes. So you can get very many different combinations of X's and Y's in spermatogenesis and many combinations of X's during oogenesis. Now what's the result of that? And I should say, like, just because I did non-disjunction up here and no disjunction up here, you could get normal separation here. You could get non-disjunction occurring at this step and this step. And so you should go through that, do a little mental exercise to figure out how many chromosomes would that mean that you could get down here. Or you could get normal disjunction here and non-disjunction here. So you can get many different combinations of chromosomes at the end products of meiosis. Now, okay, so what does this mean? So let's take some mature sperm and mature ootids. And so we're glossing over the fact that, you know, it's the secondary oocyte that is actually fertilized in humans. But we'll skip that. That's not really that important for the main point we're trying to get across in this video, which is different combinations of sex chromosomes and the effects they have on human phenotype. But let's say we had a sperm with one Y and one X. Now normally the sperm is going to be either a single Y or a single X, not both. Let's say the otid was normal. There's one X chromosome there. Well, the zygote would have one Y and two X's. And this is possible. This happens. So we could also have, say, a sperm with two Y chromosomes. And let's say the otid was also relatively normal, right? Just one X chromosome. Then we could have two Y and one X over here. How about a sperm with two X's? And again, let's go with otid, normal 1X. So you could have a zygote with three X's, zygote. How about zero sex chromosomes here? Otid, let's go with 1X. Okay, we could have 1X in the zygote. So you could do the same thing with different numbers of chromosomes, sex chromosomes in the otids, and get different combinations in the zygote. So it turns out humans can have different numbers of sex chromosomes, and these are viable. These are all humans can be viable. There are humans with different numbers of sex chromosomes. I think every combination is possible. Well, many are possible. One that isn't possible is a single Y chromosome. So all evidence so far indicates that humans cannot survive without an X chromosome. They need at least one X, and a single Y, that's not viable in humans. So essentially humans can have sex chromosomes other than either XX or XY. But with respect to other chromosomes, the autosomes, so non-sex chromosomes, humans need exactly two of each autosome, except for one of those, right? Except 21. So humans can get by with an extra 21, chromosome 21, so with 3 chromosome 21s, and that's Down syndrome. So individuals with Down syndrome have an extra number 21 chromosome. So every other autosome, if humans only have one of those or have more than 2, those are lethal. So those are lethal combinations. So humans need exactly 2 of every other autosome. I think there is, what is it, chromosome 13 and 15, I think, where humans can be born with an extra one of those, but I think they die shortly after birth. So the number of chromosomes is very, very important for essentially every autosome. Much more important than the sex chromosomes, where different numbers of sex chromosomes are usually viable in humans. And we're going to take a look at some of those next. So that's nondisjunction, and how nondisjunction can lead to different numbers of sex chromosomes, and when nondisjunction involves, also that can lead to Down syndrome, right? Nondisjunction of chromosome 21, but nondisjunction of the other chromosomes, autosomes, usually leads to a lethal genotype. Now, another thing we need to address before we talk about the different phenotypes that result from different numbers of sex chromosomes is translocation. So translocation, I should print that a little bit better here. Translocation. Now this is where, let's say, and this can involve any chromosome, and let's say this is the Y chromosome right here, the pseudo-autosomal regions, SRY, euchromatin right here, heterochromatin down here. Now translocation could involve, any part of the genome, but let's say it involves this part right here. So this part of the Y chromosome moves to another chromosome, either the X chromosome or an autosome. So a translocation is when a segment of one chromosome moves to a different chromosome. So it can involve any part of the genome, and that's called a translocation. And so we're going to see a case where translocation of the SRY to a different part of the genome can change whether or not a Y chromosome determines sex in humans. So translocation, segment of one chromosome moves to a different chromosome. So, okay, so we have this term we haven't talked about in a while, which is a karyotype. So a karyotype is the, I guess, collection of chromosomes in an individual. And the human karyotype typically involves 46 chromosomes. And when we are interested in, or we're discussing sex chromosomes, sometimes we need to specify, you know, what those sex chromosomes are. So in an individual with 46 chromosomes, and the sex chromosomes are X and Y, we would write that, you know, something like that. So we know, okay, 46 chromosomes, individual has an X and a Y chromosome. Now another possibility is 46 with 2 X chromosomes, and we could write it like that. So some common, relatively common combinations of human karyotypes are 47 with 2 X chromosomes and a Y chromosome. Also 45 with just an X chromosome. You see how the total number of chromosomes is changing? And we can tell because there's an XXY here that the difference is 1 extra sex chromosome. And here 45, okay, this individual is missing a chromosome. What chromosome is this individual missing? Because we only list 1 X here, we can see that the individual is missing an X chromosome. Another relatively common, when I say relatively common, I'm thinking like 1 in 1,000, 1 in 10,000, and I'll give you some numbers on these in a moment. So 47XYY. And then we have some other combinations where we have 46XY, but the gene, the SRY gene on the Y chromosome is mutated. So 46XY, SRY minus. So this isn't really a karyotype. This is more of a karyotype with some gene information. But I want to put this here because we're going to talk about that in a moment. So 46XY SRY plus, so a functional SRY plus, but a mutated gene called the androgen receptor. We'll talk about individuals with this genotype also. And then we also have 46XX SRY plus. So this is that translocation example we're talking about, where SRY has moved from the Y chromosome to an autosome or the X chromosome, but this is also possible, and we'll talk about this phenotype in this video. So this karyotype right here, 47XXY, okay, individuals with this karyotype are said to have Klinefelter syndrome, and I believe these are named after the physicians who identified the karyotype and associated it with a certain phenotype. So current estimates are 1 out of 660 humans have Klinefelter syndrome. And in these individuals, because there's a Y chromosome, right, we can predict, okay, there's the SRY gene is going to be present. So yeah, okay, so testes develop. And the phenotype is male. So just what we would associate with a male phenotype is seen in individuals with Klinefelter syndrome. However, and I think in the notes we have some other I guess, phenotype information about individuals with this Klinefelter syndrome. I think they're taller than normal, normal or average, taller than average, but they are infertile. So for some reason, having that extra X chromosome causes the testes to not fully develop. Let's say not fully. So they're infertile. So the sperm are not functional. So 45X, now individuals with this have Turner syndrome. Are said to have Turner syndrome. Turner syndrome is thought to occur about 1 in every 2,000 individuals. You may notice, wow, this is why is this different right here? And it might be that 45X embryos don't survive to term, as well as 47XXY. That might be the reason why we have this difference, where more humans have Klinefelter than Turner. So in Turner syndrome, so the ovaries start to develop, right? Because there's no SRY gene. Ovaries start to develop, but then something goes wrong. And scientists think now, during embryogenesis in humans, as soon as the ovaries start developing, the oocytes start to enter meiosis, right? And it might be the failure of the X chromosome here to pair with another X chromosome that causes meiosis to abort, and then that feedbacks and causes the ovaries that somehow aborted meiosis causes the ovaries to stop developing. And this is where we get the streak gonads developing. So Turner syndrome, so the overall phenotype is female, external genitalia is female, but they might need hormone replacement therapy. This is a very complicated subject. So hormone replacement therapy, because the gonads don't produce the streak gonads don't produce the hormones that would normally be produced by the ovaries. So 47XYY. Now these this is called the super Y, or one karyotype, and essentially these are, the phenotype is really no different from, I think they might be taller than average than normal 46XY males, but otherwise normal male phenotype, what we'd associate with a normal male phenotype. And it occurs about 1 in 10,000 males, so that's the incidence rate. And in the 1960s, something interesting is that scientists did studies, and they found that, you know, if the super Y karyotype is found at 0.1% in the general population, they looked at males that were, I guess, in either institutionalized males, so they were in either a mental institution or the prison system, or sometimes a mental institution and a prison system, and they found that rates were as high as 2%. So that led to this hypothesis that males with 47XYY genotypes were predisposed to antisocial behavior. And that type of behavior got them institutionalized. So in the 1980s, there was a large grant from the US government to follow 20 infants that were born with this karyotype, and, you know, compare them to infants without this karyotype and see is it true, you know, are they prone to antisocial behavior? And so that study was ended because once the public found out about that, rightly so, right? They terminated funding for that project, because, you know, how would you like to be the focus of a bunch of scientists thinking you are predisposed to an antisocial behavior and they're waiting for you to do something wrong, you know? So there's lots of ethical things wrong I mean, I think there are lots of ethical things wrong with that type of experiment, and clearly a lot of other people did, so they ended it. And so now what we think is, it seems like there might be a slightly higher tendency towards antisocial behavior if you're a male with 2 Y chromosomes. But we don't really know for sure. Again, this was a study in the 60s, and there was a lot of interest in this, and I haven't seen any new studies on this, so we'll just leave it at that. So, okay, so let's take a look at this one, 46XY SRY minus. Now, okay, so the SRY gene is mutated. So what do we think is going to happen? So testes are not going to develop. So let me put this right here, 46XY SRY minus. So individuals with this disorder have Swire syndrome, or this genotype, I should say. Now, maybe some of you might be confused. What's a karyotype? What's a genotype? So that's, say, a karyotype, but when I have to throw in specific genes, let's call it a genotype. We'll call it a genotype if we have to indicate a specific, mutation of a specific gene. So Swire syndrome. Okay, so this is called Swire syndrome, and what happens in Swire syndrome, let me see. So testes do not develop, right? We can predict that because the SRY is mutated, and SRY provides the signal for the medulla to develop into testes. Testes do not develop. And just like Turner syndrome, there's only one X here. There's only one X. But okay, so what is this? But what happens also is that streak gonads develop, and it's not exactly like Turner syndrome, right? Because if in Turner syndrome, it's the failure of the X to pair during meiosis that leads to formation of the streak gonads. Well, the X can pair with the Y, so it's not going to be quite the same. And I'm not even sure scientists have quite figured out why the streak gonads develop in this case. Why don't ovaries fully develop? But they don't develop. So streak gonads develop. So these individuals are usually raised as girls or with a female gender identity, normally female. So the external genitalia are female, etc. But they do need hormone replacement therapy, right? Because streak gonads instead of ovaries develop. So hormone replacement therapy is generally used in individuals with Swire syndrome. So again, why the phenotype of Swire is seems significantly different from the phenotype of Turner. I'm not really sure. Those of you who want to go to medical school, it'd be OB/GYNs or pediatricians, might have to focus more on this in the future. But it seems to be it's relatively very interesting subject, and I know there's a lot of research being done on the types of hormone replacement therapy needed for individuals with Turner syndrome or Swire syndrome. Now we have 46XY SRY plus. Let's see, and AR minus. Okay, so what is this? Now, with this genotype right here, you don't know what AR is yet, but so with this genotype, we see 46XY SRY plus. So this is just normal male, genetic male genotype. But then we put this in here, AR minus. What is that? So AR is the androgen receptor. Now, the presence of SRY leads to the development of testes. And the testes make testosterone. But this thing right here, the androgen receptor, is a protein that's required for recognizing testosterone. So AR, the androgen receptor, recognizes testosterone and then signals, let's say triggers, say the male secondary sex characteristics. So if this is mutated, well, there's testosterone present, but the testosterone can't trigger the male secondary sex characteristics because it does it through the androgen receptor. So what happens is the testosterone breaks down into estrogen. And the presence of estrogen triggers female secondary sex characteristics. So the phenotype of these individuals is typical of what you'd associate with just a normal, I have screen here, just a normal genetic female, except that in place of ovaries, there will be testes present. However, the testes do not produce functional sperm. And the reason they don't produce functional sperm is because the androgen receptor and testosterone have a role in making functional sperm. So the fact that that's not there, the testes are there, they're making testosterone, but they don't make functional sperm. So individuals with this are phenotypically female, but they have testes and they are infertile. And I believe in the notes, I have some links to, I guess, a young woman from, well, she's probably in her 30s now, who talks about her experience with, having this genotype right here. And the syndrome is called Complete Androgen Insensitivity Syndrome, CAS. And it occurs about 1 in 20,000 humans. I think this is 1 in 20,000 humans, not 1 in 20,000 females. Yeah, let's just say this is 1 in 20,000 humans. Okay, so one more. So we have 46XX SRY plus. Okay, so if you saw this genotype here, first, what would we say? How is this possible? So, this must be translocation. So the SRY gene must have moved either maybe during meiosis, sometime during in the germline of one of the parents. The SRY gene moved, let's say, from during spermatogenesis to an autosome, and now we have offspring with this genotype, 46XX SRY plus. So it turns out the normal phenotype here, this is a phenotype, male phenotype, so this is all the secondary sex characteristics are those we would attribute to a genetic male, so XY male, except the, let's see, testes do not fully develop. So they're infertile. Now there are lots of genes on the Y chromosome that are necessary for spermatogenesis and formation of functional sperm, so it makes sense that an individual with this genotype right here, lacking all those other genes in the Y chromosome, would be infertile. And this happens about 1 in every 25,000. The frequency is about 1 in every 25,000 humans. Okay, so that's it for the different, I'm sure there's lots of others